Hi everyone, I'm Jessica VASU. My matrix number is 23DBT20F2019 and I'm from Polytechnic Nilai Negeri Sembilan. Today, we are going to see about Pcr mechanism for my DYB 30093 molecular biology subject. So let's get started. Pcr is a powerful lab technique used to generate billions of copies of specific sequence of DNA. Having many copies of particular sequence allows you to read, compare, and manipulate them, which is useful in many fields. To amplify a certain region of DNA in all Pcr reactions require a buffer. The reaction takes place in Pcr buffer with optimal pH and salt concentration that ensure maximum Pcr yield. Magnesium ions are an important part of the buffer as they help to stabilize DNA and activate the DNA synthesizing enzyme Taq polymerase. Secondly, we need a template DNA. A DNA template, which is a stretch of double-stranded DNA containing the region we want to amplify. The two strands of the DNA template have the opposite orientation relative to each other. The 5 and 3 ends are opposing each other and interact with each other by complementary hydrogen bonding. Next, we need a set of primers. Primers or oligos are short pieces of single-stranded DNA that are complementary to the ends of the DNA region that we want to amplify. Primers are necessary in order to Taq polymerase to create a new strand of DNA. Where primers attach determines what region is amplified. Next, we need DNTPs. The building block of DNA, the 4-deoxyribonucleoside thyrophosphates, DNTPs, are DATP, DCTP, DGTP, and DTTP. These DNTPs are provided in huge excess in a Pcr reaction. Next, we need Taq polymerase. A special DNA polymerase called Taq polymerase synthesizes DNA by using DNTPs to elongate the primers as it uses single-stranded DNA as its template. Unlike DNA polymerase found in eukaryotic cells, Taq polymerase is heat resistant, allowing it to function at the high temperature used in Pcr. Now all Pcr regions are combined in the Pcr tube in a buffer that ensures most Pcr product. Magnesium and potassium ion concentration are particularly important and sometimes need to be adjusted. With the reactants in place, the Pcr tube is ready for the thermocycler where the Pcr cycles can begin. One Pcr cycle consists of 3 steps: denaturating, annealing, and elongation. This cycle will be repeated again and again to create a huge number of copies of the same specific DNA segment. Let's look at the first Pcl cycle, denaturation. During denaturation step, the PCR reaction mixture is heated to 95 degrees Celsius, breaking the hydrogen bonds between the DNA strands. At this temperature, all DNA exists as single strands. Depending on the exact sequence of the DNA template, the dissociation of the double-stranded DNA into single strands occurs at different temperature. However, at 95 degrees Celsius, all DNA will be single-stranded. After denaturation, the temperature in the PCR tube is lowered to allow the primers to anneal or bind to their complementary regions on the single-stranded DNA templates. Depending on the sequences of the primers, the annealing occurs between 45 to 70 degrees Celsius. The annealing temperature for each primer set is different due to the number of hydrogen bonds formed with the DNA template and has to be calculated individually. The short primer-base pair to the template with great specificity so that two template primers complexes are formed. Primers are supplied in great excess and will not be depleted during the PCR reaction. During elongation, the temperature is raised to 72 degrees Celsius, which is the ideal working temperature for Taq polymerase. The polymerase uses annealed primers as the starting point of DNA synthesis. Each primer is extended along its DNA template by addition of complementary DNTPs to the 5 to 3 direction until the end of the DNA template. The result of the first PCR cycle is a copy of the region to amplify with an added overhang. Future PCR cycles will not generate these added overhang regions. As we will see, after 30 cycles, only region of interest will be exponentially amplified. For the second cycle, we go through denaturation, annealing and elongation again. After denaturation, both the original strands and the two newly made copies with the overhang serve as templates for the primers to anneal. After primer annealing, the Taq polymerase extends the primers and synthesizes new DNA along the 4 templates. The result of the second cycle is 4 pieces of double-stranded DNA. Most of the DNA strands contains overhangs, but the 2 most recently made strands are the correct length. They consist of the region to amplify. In the third cycle of PCR, these 4 double-stranded DNA molecules provide 8 templates in the denaturation phase. The temperature is reduced to 60 degrees Celsius and the primers anneal to the 8 templates. The temperature is raised to 72 degrees Celsius and Taq polymerase extends the annealed primers along the templates. After going through the PCR cycle a third time, we will end up with 8 double-stranded PCR products. 2 of them finally exactly match the double-stranded DNA segment we wanted to amplify. 4 more strands of DNA have the correct length of the region to amplify. In the following cycles of PCR, the desired product will be enriched exponentially. Typically, a PCR protocol will repeat 30 cycles cycles of Pcr produce 30 cycles of denaturation, annealing, and elongation. 30 cycles of Pcr produce around 1 billion copies of the amplified region over only a few overhang products. As we can see, these are the length and the desired region of cycles, in which we can see this number of cycles here. After 30 cycles, the temperature is held for 10 minutes at 72 degrees Celsius to ensure complete synthesis of all Pcr products. Finally, the temperature drops to 4 degrees Celsius to refrigerate the sample. The final Pcr product can be visualized by DNA gel electrophoresis. This technique uses electrical charge to separate DNA molecules based on their size. If the Pcr reaction was successful, the Pcr product appears as a single band in the DNA gel because virtually of all the DNA molecules generated at the same time. In this process of combining target DNA, primers, dNTPs, and tag polymerase in a buffer, and then cycling 30 times through predetermined thermal cycles, we can generate more than a billion copies to amplify from just a single copy of our template DNA. This is the power of polymerase chain reaction. That's all for today. Thank you very much for watching.